Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, inshallah today we're going to discuss the central nervous system, the second chapter in the module of neuroscience 205. Here we have levels in the CNS, we have levels in the spinal cord, medulla, midbrain, pons, and we have cerebrum and cerebellum. Light microscope, we have section for the cerebrum and for the cerebellum, low power slide, and we're going to have high power slide of the cerebellum. First, we are going to discuss the levels. First, we're going to have the spinal cord. As you can see here, dear students, as you know that the central nervous system is formed of brain and spinal cord. The spinal cord is formed of 31 segment, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. We have TS section in the spinal cord. I'm going to discuss 3 levels, the cervical, thoracic, and the lumbar. First of all, from where I know that this is the spinal cord? From what? From the H-shaped gray matter. H-shaped gray matter. Okay? I'm having in the center central canal lined by simple cuboidal epithelium, partially ciliated, which is known as the ependymal cell. We have the peripheral white matter, and the H-shaped gray matter, In the center, I'm having one, this is posterior and this is anterior. The posterior median septum, and anterior median fissure, The gray matter is formed of nucleus and unmyelinated nerve fibers and neuroglia. We have 2 dorsal or posterior horns which contain sensory nuclei. I'm having 2 posterior and 2 anterior or ventral horns containing motor nuclei. Okay, the white matter is formed of myelinated nerve fibers and neuroglia. Each half of the spinal cord section, I'm going to divide it like this. Okay, I'm going to divide it like this. I'm having 3 columns. One known as posterior column, present between the posterior median fissure, present between the posterior median septum, and the posterior horn. Then we have the lateral column. Present between the anterior horn and the posterior horn. This is the lateral column, present between the posterior column and the anterior column. Then we have the anterior column, present between the anterior horn and the anterior median fissure. This is the white matter. So now we know that we are in the spinal cord. What level? I'm having 3 levels, which are the cervical, thoracic, and the lumbar. This is the cervical. From where you know? First, look at the outline. Look at the outline. Appears oval. Then, look at the posterior horn. Appears thin and divergent. Look at the anterior horn. Appears thick. Look at the white matter. More white matter than the gray matter. And the central canal appears more anterior. Again, what are the criteria? First, the outline appears a little bit oval. Second, the posterior horn thin and divergent. Third, the anterior horn thick. Fourth, more white matter. Number 5, the central canal more anterior. Okay, dear students, the second section in the spinal cord thoracic segment. First, why it is spinal cord? H-shaped gray matter. Why it is thoracic segment? Look here at the posterior horn appears thin and little bit parallel to each other. Thin and parallel to each other. As well as the anterior horn appear thin and parallel to each other. The central canal appears more central, and the white matter start to be less than that of the cervical segment. Again, why it is spinal cord? H-shaped gray matter. Why it is thoracic? Both the posterior horn and the anterior horn are thin. The posterior horns are parallel to each other, as well as the anterior horn. The central canal more central, and the white matter start to decrease. Again, dear students, this is spinal cord. Why? H-shaped gray matter. What level? Lumbar. Why? Look at the posterior horn is thick and parallel to each other. Look to the anterior horn, thick as well as parallel to each other. The central canal more anterior. The white matter decrease a lot. Here I can see the posterior column between the posterior median septum and the posterior horn. The lateral column between the posterior horn and the anterior horn, while the anterior column between the median anterior fissure and the anterior horn. Lumbar segment. Why it is lumbar segment? Thick parallel anterior and posterior horn, and decrease white matter, as well as the central canal more anterior. What is this section, dear students? It is closed medulla. Closed medulla. What level? Motor decussation. Closed medulla, motor decussation. This is a section in the medulla. Why we said it's closed? Because I can see central canal. Why it is motor decussation? Look here, dear students. This is the gracile nucleus, this is the cuneate nucleus, and this is the spinal nucleus of the trigeminal nerve. The gracile and cuneate nucleus are small, while the gracile and cuneate tract is big. Here, more anteriorly, there is the direct pyramidal on both sides of the medial line here. Closed medulla, motor decussation. Both the gracile and nucleus small, while the tracts are large, and there is the spinal nucleus and tract of the trigeminal nerve. On both sides of the median line here, anteriorly, is the direct pyramidal tract. Here, what this, dear students? It is again closed medulla. Why? Because I can see in the middle line the central canal. Sensory decussation. Why? The gracile and cunate nucleus increase and their tract decrease, as well as this is the spinal trigeminal nerve and tract. On both sides of the median line here, we have the pyramidal tract and the olivary nucleus. Closed medulla, sensory decussation. Why it is closed? Due to presence of the central canal. Why it is sensory? Both the gracile and cunate increase, the nuclei become bigger, and the tract is less. And this is the spinal trigeminal tract and nucleus. More anteriorly, there's the pyramids. Above them is the olivary nucleus. This section, dear students, is the pons. Okay, I can see in the middle line no opening, no central canal. Here is the fourth ventricle. This group is the fourth ventricle. And what is this part of the cerebellum? And on both the sides there are the inferior cerebellar peduncles. And here is the basis pontis. The midbrain, dear students, is divided into three parts. The tectum, which contain either superior colliculus or inferior colliculus. The tegmentum contains many components. The most important is the aqueduct of Sylvius and the gray matter surrounding it. And the basis pedunculi contains many fibers. Medially, there is the medial corticopulbar and corticopontine. Laterally, lateral corticopontine and corticopulbar, and in the middle is the corticospinal fibers. So how could I differentiate between the superior and inferior colliculus? Look at the tectum here. Look at the tectum here. Okay, here. Okay, the colliculus, if this is the middle line, you can see it appears like a concavity here, the superior colliculus. The aqueduct of Sylvius, appears to be pear-shaped, as well as the gray matter surrounding it, inverted pear-shaped gray matter. I can see also in the tegmentum the red nucleus. I can see the red nucleus. Okay, appears as two bulges here, the red nucleus. Okay, so this is the midbrain superior colliculus. Here is the midbrain inferior colliculus. I'm going to divide them as well into the tectum. In the tectum here, I can see the inferior colliculus. Appears as one oval mass. In the tegmentum, I can see the aqueduct of Sylvius. Here. The aqueduct of Sylvius, surrounded with quadrilateral shaped gray matter. Appears as if it is oval or rhomboid, surrounded with the quadrilateral or oval gray matter. Then here we have the basis pedunculi. So why it is inferior colliculus? Look at the tectum. You can see it as a one oval mass. Look at the aqueduct of Sylvius, appears oval, surrounded with oval or quadrilateral area of gray matter. And then this is the basis pedunculi. Here, dear students, the cerebrum or the cerebral cortex. You can see here the slide in the data show and under the microscope. The cerebrum is formed of sulci and gyri. The outer part is dark gray matter, and it's called the cerebral cortex. Here, the gray matter is different than that of the spinal cord. The gray matter in the spinal cord was central. Here, the gray matter is at the periphery, at the cortex. The gray matter appears dark and present at the peripheral part, which is the cortex, while the inner is the medulla, which appears pale. So again, the cerebrum or the cerebral cortex, you can see the section under the light microscope and in the data show slide. Sulci and gyri. Outer part is dark, called the gray matter of the cortex. Inner part is pale and called the white matter or the medulla. Here, dear students, is the cerebellum. Cerebellum. It appears folded. This folds is called the folia. Each folia is formed of outer dark gray matter called the cortex. Where, dear students? Here. All this known as the cortex. Okay? Inner part here, the inner part here is the pale medulla. So the cortex is that. Here is the cortex. Inside is the medulla. Higher magnification, dear students, on the cerebellum. I can see here what this. It is the outer cortex, and this is the inner medulla. Again, all this is the cortex, and this is the medulla. The cortex is formed of the following layers. Molecular layer, Purkinje layer, and granular layer. Again, it's formed of molecular layer from outside. Then we have the Purkinje layer, then the granular layer. All this is the cortex. Here is the medulla. Higher magnification. Another higher magnification of the cerebellum. Here is part of the cerebellar cortex formed of 3 layers: molecular layer, Purkinje layer, and granular layer. You can see here the Purkinje cell is piriform multipolar in the process of large extending to the molecular layer. Again, dear students, higher magnification of the cerebellar cortex. This is the molecular layer, Purkinje layer, and granular layer. As you can see here, the Purkinje cells are large, piriform multipolar, arranged in rows. Again, dear students, this is the cerebellar cortex, molecular layer, Purkinje cell layer, and the granular layer. As we said before, the levels, data show slides, while the cerebrum and cerebellum data show and light microscopic low power, and the Purkinje cells high power under microscope. Now we were going to do the trial exam. Okay. Now, dear students, what is this? It is spinal cord. Why? Due to the presence of the H-shaped gray matter. What level? The lumbar. Why? The posterior horn thick and parallel, as well as the anterior horn, the central canal more anteriorly, and the white matter decrease a lot. Number 1 is what? Is the lateral column. White matter between the posterior horn and anterior horn. Number 2 is what? Is the anterior median fissure. Anterior median fissure. Here, dear students, what this section is. It's not spinal cord, yes? Okay, I can see here central canal. I can see gracile and cunate nuclei large. So it is closed medulla sensory decussation. Gracile and cunate nuclei, spinal trigeminal. Here is the pyramid and here is the olivary nucleus. So this level is closed medulla sensory decussation. Number 1 is the number 1 is the spinal trigeminal. Number 2 is the medial accessory olivary nucleus. Okay, dear students, what is this? It is midbrain inferior colliculus. Why? Look at the tectum. I can see oval mass, which is the inferior colliculus. Here, number 2, the aqueduct of Sylvius, surrounded with quadrilateral or oval gray matter. Here, dear students, is the pons. I can see here the fourth ventricle. Here is the cerebellum. Number 1 is the inferior cerebellar peduncle, and number 2 is the basis pontus. What this section, dear students? It is spinal cord. Why? H-shaped gray matter. What level? Cervical. Why? The posterior horns are thin and divergent. Number 2 is the central canal. Number 1 is the anterior horn. As you can see here, there is much white matter. What this level, dear students? It is closed medulla. Why? I can see the central canal. Motor or sensory? Motor. Why? The gracile and cunate nucleus is small, and I can see the direct pyramidal. Number 2 is the cunate. Number 2, the cunate nucleus. This is the gracile, cunate, spinal trigeminal. Number 2 is the cunate. Number 1, on both sides of the median line here, we have the direct pyramidal. Closed medulla motor decussation. Here is the midbrain superior colliculus. Why? Look here at the tectum. I can see the superior colliculus. Number 3, superior colliculus. Here I can see in the tegmentum the aqueduct of Sylvius. Number 4, surrounded with pear-shaped gray matter. Number 2. As well, I can see the red nucleus. Okay. Mention one visible feature for 2. Here I can see an inverted pear-shaped appearance of the gray matter. So it is midbrain superior colliculus. Here is the tectum. Number 3 is the superior colliculus. In the tegmentum, I can see the aqueduct of Sylvius. Number 4, surrounded with inverted pear-shaped gray matter. Both sides here, I can see the red nucleus. Number 1. Dear students, is a section in the cerebellum. From where I know, from the folia, this fold. Number one is the pale white matter. Number two is the dark gray matter. The dark gray matter, or the outer cortex that's formed of the molecular Purkinje and granular layer. To summarize, these students, the data show in the microscopic slide for the CNS, the levels including the spinal cord levels, cervical, lumbar, and thoracic, medulla, closed motor and sensory decussation, midbrain superior inferior colliculus, the cerebral cortex and the cerebellum, and including high power for the cerebellar cortex and their layers, as well as the Purkinje layer. For the light microscope, we have only the cerebrum cerebellum. High microscope, we have the Purkinje cells. Okay, thank you, dear students.